Hey everyone, and welcome. So, we've all heard that we get our treats from our parents, right? But how does that actually work? Why do some features show up while others seem to just disappear? Well, today we're going to dive into one of the most fundamental rules of genetics that's going to unlock some of the biggest secrets hidden in your DNA. Have you ever looked in the mirror and wondered, why do I have my mom's eyes but my dad's hair? It's definitely not just a coincidence. There's a powerful principle of genetics at play here. And you know what's crazy? The whole story, the key to understanding this, actually started in a simple garden with some pea plants.
So let's meet the man himself, Gregor Mendel, who we now call the father of genetics. Way back in the 1860s, this monk, working quietly in his monastery garden, started doing some experiments with pea plants. And in doing so, he cracked a puzzle that had stumped people for centuries. How exactly are traits passed down from one generation to the next?
But here's the thing. Mendel's work wasn't an overnight sensation. Not at all. In fact, it took decades for the rest of the world to catch up and realize his genius. It wasn't until the early 1900s, when his papers were rediscovered, that the foundation for modern genetics was truly laid. And from there, things really took off. We got tools like the Punnett square, and scientists began to debate just how these principles actually worked in the real world. So, here was the big mystery that Mendel was staring at. When he crossed a tall pea plant with a dwarf one, all the offspring in the first generation came out tall, every single one. The dwarf trait, it just seemed to vanish into thin air. But, and this is the crucial part, when he then crossed those tall plants with each other, that dwarf trait suddenly popped right back up in the next generation. This meant that traits weren't just blending together like paint. No, something else was going on. They were following some kind of hidden pattern. And the principle that Mendel came up with to solve this mystery, well, it was nothing short of revolutionary. He called it the rule of dominance. So let's get right into it and crack the code of what this really means. Okay, so complete dominance is what happens when for any given trait you have two different versions of a gene. We call these versions alleles. Think of it like having two different sets of instructions for the same job, but only one of them gets read out loud. The allele that gets expressed, the one you actually see, is called the dominant allele. And the one that gets hidden, that's the recessive allele. Now, this brings us to two super important terms you've got to know: genotype and phenotype. The genotype is the actual genetic code an organism carries. It's the set of alleles it has, like TT or TT for plant height. The phenotype, on the other hand, is the physical trait that you can actually see, like the plant being tall. So here's the key takeaway. You can have the same phenotype, a tall plant, that's actually hiding two different possible genotypes. Just take a look at this example. It makes everything crystal clear. When you take a pure red tulip, which has two dominant alleles for red, let's call them big R, and you cross it with a pure white tulip with two recessive alleles, little r, all of the offspring come out red. Why? Because that one dominant red allele completely masks the effect of the white one. So how can we actually predict the results of these genetic crosses? Well, this is where a really handy tool comes into play, called a Punnett square. It's basically just a simple grid that helps us visualize all the possible outcomes when we combine genes. All right, let's go back to Mendel's classic experiment. Take a look at this diagram. He started with what's called the P generation, for parental. This was a pure-breeding tall plant, TT, and a pure-breeding dwarf plant, TT. When he crossed them, the next generation, the F1 generation, were all tall, every single one. Their genotype was TT, and that dwarf trait was completely hidden from view. Okay, now here's where it gets really cool. What happens when we cross two of those F1 plants together, both with the TT genotype? We use a Punnett square. The first step is to take the possible gametes from each parent, in this case, a T and a T, and list them along the top and the side of the grid. Then you just fill in the boxes by combining the letters. It's that simple. And voila, here's the result. This finished grid shows us all the possible genotypes for that second or F2 generation. We get one TT, two TT, and one little t. That gives us a genotypic ratio of 1 to 2 to 1. So you have one pure breeding tall, two hybrid talls, and look at that. The pure breeding dwarf plant made a comeback.
But what if we just look at what the plants look like, their phenotype? Well, the story changes a little bit because the tall T allele is dominant. Both the TT and the TT plants are going to look tall, only the TT plant will actually be dwarf. So what that gives us is a ratio of 3 tall plants for every 1 dwarf plant. And that right there is Mendel's famous 3-to-1 phenotypic ratio. Now, complete dominance is a fantastic rule, but nature doesn't always play by such simple rules. Sometimes things get a little more complicated, and that's when genetics gets really interesting. So let's take a quick look at a couple of the exceptions. Okay, now pay close attention to this. On the left, you've got the complete dominance we just talked about. Red plus white equals red. But look at the right side. Something totally different is happening. This is called incomplete dominance. In some flowers, like snapdragons, when you cross a red one with a white one, the offspring actually turn out pink. In this case, neither allele is truly dominant. Instead, they blend together to create an entirely new intermediate phenotype. And then there's one more fascinating case called codominance. Now think of this as both alleles showing up to the party and being expressed fully and separately. They don't blend. The perfect example of this is the human ABO blood group system. If you inherit an allele for A-type blood and an allele for B-type blood, you don't get some blend, you get AB blood type. Both traits are expressed at the same time. So, from a humble garden full of pea plants all the way to the complex code of our own DNA, these principles that Mendel discovered are just as important today as they were back then. They are the foundation for understanding who we are, why we are the way we are, and how this beautiful, intricate dance of life gets passed on from one generation to the next. All right, let's do a super quick recap of everything we've covered. First, traits are passed down through what we now call genes. These genes have different versions, or alleles, which can be dominant or recessive. In complete dominance, the dominant allele completely masks the recessive one. Remember, genotype is the genetic blueprint, while phenotype is the physical result. And finally, the Punnett square is our tool for predicting the probability of those outcomes, like that classic 3-to-1 phenotypic ratio. Mendel's simple rules really threw open the doors to the entire field of genetics, paving the way for everything from understanding hereditary diseases to mapping human evolution. So the next time you look in the mirror, just take a moment and think, what other stories are your genes telling about you, just waiting to be discovered? Thanks for joining us.